very much, Chris. And I guess I'm very fortunate from two counts this morning. As I say, I'd come down from Scotland last night on the sleeper. We haven't yet got spring in Scotland. It's nice to see green leaves. It was snowing in Edinburgh yesterday afternoon. Secondly, and more seriously, it's great to follow Fiona's talk. As a fellow biochemist, thank you very much, Fiona. I'm glad there's at least two of us in the room. But more seriously, you raised the point about the importance of enabling collaboration across industry, NHS and academia to deliver this capability. I'm getting some feedback here. I'm going to drop this down. Is that any better?
Yes.
Good. So I'm going to give an overview of an exciting initiative we've been running for the last 3 years in Scotland called Stratified Medicine Scotland Innovation Centre. With particular reference, because it's an informatics talk, to a multiple sclerosis project we're running, and the reason for using that as an exemplar is to describe a sub-project within that to enable interoperability between NHS regular clinical records and our own research needs, and how we're streamlining that with a view to making this standard way of engagement. So a quick overview, what on earth is this SMSIC thing? As I said, it's been going for 3 years now. It's a 10-way collaboration of 2 industrial partners, 4 NHS health boards, and 4 universities across Edinburgh, Glasgow, Dundee, and Aberdeen. That covers roughly 83% of the Scottish population, and it's my job to manage that lot. So some of my best friends are lawyers, and you can imagine getting 10 sets of lawyers in a room at any one time to agree anything other than what day of the week it is quite fun. So we've been going for 3 years. The aim is to set up an industry-facing capability in stratified medicine. So this is very clearly not an academic research centre. This is about providing national capability to start answering questions from industry, particularly around pharmacogenomics or disease progression. We have 2 industrial partners in the shape of Thermo Fisher Scientific, whose next-generation sequencing platforms we use, and then Iridia Informatics, who provided much of the high-performance compute capability and compute consultancy that has allowed us to set up this collaboration. We're based currently in the new Queen Elizabeth University Hospital in Glasgow, so a £1 billion development opened last May. I think it's the biggest single critical care complex in Europe, something like 1,100 beds and 20 theatres, and that serves something like 2.3 million of the Scottish population. It's even got a helicopter pad on the roof, so if you fall off a mountain in the Isle of Skye, you could be in the hospital in about 10 minutes, I believe, though I wouldn't want to try it. So we're based in there, which is great. You may ask the question, why would we do precision medicine in Scotland? This is a slide I put together in my Wyeth days, in pharma industry days, and I've kept on using it because it kind of works. The Scottish population is a 5.3 million population, so it's about 8% of the UK. The good thing is, from a stratified medicine perspective, they are geographically static, so incomers and outgoers are pretty low, so it's great if you want to do longitudinal studies. We're also pretty unhealthy. You know, I usually get a giggle from the audience about this. You know, most of the stereotypes are broadly true. I won't go into them, we all know them. But more importantly, the level of unhealthiness is very well characterised electronically. So Scotland, back in the 1970s, set up this unique patient identifier system called the Community Healthcare Index, or the CHI number, which basically populates every one of those NHS databases across multiple health boards across Scotland. So if you really want to discover which John Smith you're dealing with, you can find him. And you've got a unique identifier for John Smith across all touch points with NHS records. So that's really helpful. And the final point is the Scottish population on the whole has proved very well disposed to getting involved in clinical research, which is really helpful. Going back to my previous assertion about we're an unhealthy lot, this is the graph that proves it. Now all the audience is going to be looking at, where do I fit on this graph? This is the incidence of chronic disease across that 5.3 million patient by, I think, 5-year increments there. And so the take-home message is, by the age of 50, more than half the Scottish population has got at least one chronic disease, and by the age of 65, I believe we've got at least, more than half the population got at least 2. So that kind of substantiates my claim. Going back to the extent and preparedness of the patient population to get involved, this is an exciting initiative that is really supportive of stratified medicine in Scotland. Just to be clear, this is not me. This is an initiative called SHARE that was set up about a year ago. As you can see, the registrations in SHARE now is somewhere north of 100,000 people. All this is an online pre-consenting capability such that as an NHS Scotland patient, if I consent to this, what I'm doing is saying if I go into the NHS for any routine procedure, biopsy, blood involves blood collection, biopsy, any spare material that is left over from whatever they want to measure me is pre-consented for storage, pre-consented for research purposes. I have further consented to include genetic sequencing in that, and I have further consented to allow contact by industry through PIs for potential involvement in clinical studies. It's a fantastic resource. As you can imagine, as that 100,000 patient population gets older and picks up the usual collection of chronic disease, you start having before and after samples in there for whatever chronic disease you've got. The intent is to build this to about 500,000 base, which is 10% of the population. So it's going to become a very, very powerful resource, and it's proving extremely popular. As I mentioned, the NHS Scotland has multiple healthcare database systems. If you like, the womb to tomb strapline seems to work, and as you progress through life, you populate most of that lot. The point with the Chi record, the Chi number, is pulling records from across combinations of these and from across multiple health boards is much more tractable than elsewhere. So again, fantastic tool for clinical research. My own team's deployed the Thermo Fisher Life Technologies ion torrent sequencing platforms. Sure, this audience is well familiar with those. We've had a fantastic amount of support from Thermo Fisher over the last 2.5 years in getting these set up, and can support the usual combination of gene panel, exome, whole genome, various modalities of RNA sequencing. Not going to be talking about that today, but happy to go offline if anyone's particularly interested in it, but they've been a great partner. In terms of the informatics, getting this right is key, especially when you're running multi-partner collaborations. You've got gangs of people spread across multiple universities, multiple health boards, SMEs, the likes of Thermo Fisher, who all need to look at, contribute to phenotype and genotype data in a research analytics environment. So providing a stable, professionally supported platform to do that is extremely important. And we've done a lot of work with Iridia over the last 2 years to really prove this out. So that's our HPC cluster in the Queen Elizabeth Hospital. And we're proving that that's being extremely useful platform for doing what we need to do. We're also finding that there's a market for this and effectively contracting out this capability to other collaborations outside our own who just need access to an HPC capability for the 2 years or 18 months of their project with a couple of dozen players across the world. So that's proving an interesting sideline for us. What we've been setting up, as I mentioned, this is industry facing. We have used some of our initial runway funding to get a number of what we call exemplar projects up and going, mainly in cancer, rheumatoid arthritis, multiple sclerosis, which I'll talk about. And these are around proving out the basic capabilities you need in this complex collaboration structure around managing projects, supporting next-gen sequencing, having the ability to process and support phenotype and genotype data sets and the analysis thereof, and coming back to it, integration with healthcare data sets. So I'm going to be talking about a project called Future MS, which we've undertaken with a gent called Professor Siddharth Chandran out at the Anne Rowling Clinic in Edinburgh. This is a national initiative across Scotland. Those of you who know something about Scottish patient predispositions to disease will know that MS is more frequent, more prevalent in Scotland than anywhere else in the UK. And interestingly, as you go further north in Scotland, the worse it gets. So I believe the incidence rate in the sort of central belt, Glasgow, Edinburgh region is about 3 times the home counties in England. And as you go up to Ullapool in the north, it goes up and at another 2-fold. Don't really understand the reasons for it. The genetic component is thought to be around about 30%, but no more. It may be lifestyle, it may be lack of sunlight, we don't know. There are parallels in Scandinavia, Norway, and Sweden, but the real basis for it is not really understood. 2.3 million cases globally, about 110,000 in Scotland. As you know, it's fairly devastating. The difficult piece from the neurologist perspective is the freshly newly diagnosed MS patient. We have very little evidence base to predict what the disease trajectory for that patient is going to be. Are you going to be one of the relapsing remitting patients who can broadly carry on reasonably as is, if you like, for 10, 15, 20 years, or are you one of the progressive forms of MS, which could mean in worst case you're in a wheelchair in 5 years from diagnosis? And at the point we are today, we don't have the evidence base to say which of these tracks are you following. This obviously affects the patients big time because they'd really like to know this. They'd like to be able to plan for the future in whatever shape or form that takes. Also, from a disease management perspective, you'd like to know who you're going to want to prescribe the expensive biologicals at £50,000 a therapy to. Really, you probably want to be targeting these folks if only you knew who they were. So there's a real opportunity to make some real positive gains here. Where we're going with the project is saying, can we recruit an inception cohort across Scotland over the next couple of years, about 500 patients. We obviously don't know what their disease trajectory is. That's why they're all black up here. At baseline, we'll be collecting an MRI scan, full patient CRF, a number of lab data sets, undertake some sequencing, follow the patient through the first 12 months of their disease, get them back in for a second visit, repeat the MRI scan, and build out further on the CRF data set. What that allows you to do then is compare brain volume and lesion counts from MRI at T1 year and baseline and say who's got worse over that period and who hasn't. And then the intent is that we can then pull in the sequencing data sets and the labs and CRFs and say, can we therefore say on the basis of the MRI data whether you are likely to be in the remission set or the progressive set? Now, if we're fortunate with this, we'll then have, if you like, a genetic test possibly coupled with some biomarker tests that allows us to take future newly diagnosed patients and say which of these tracks are you likely to be going through. So that's the high-level study plan. Just starting off right now, as I said, running across Scotland, there is a fairly complex set of logistics around where we're running the clinics, where we handle sample processing, sample extraction, sample storage, bioassay sequencing, and ultimately routing all that data into our research analytics hub to allow us to look at these phenotype-genotype relationships. So if you like, the challenge for us as a project is we're going to be obviously working with normal standard of care activities within the NHS. They're going to be generating significant amounts of routine care data sets coming out of these 4 health boards. How can we engineer appropriate interoperability here so that we can read out those data into our research analytics cluster down here in an appropriate manner without near enough in real time? So that's pretty much saying the same thing. What we're really interested in this particular case for the purpose of this presentation is not the genomics data that we're handling quite nicely, but how can we access the 8 or so different lab sets of data here, vitamin D levels, cholesterol, lipids, liver function tests, etc., for the patients who are consented to this study from those 4 NHS boards. So again, I'm a biochemist giving an informatics talk. I'm not going to go too heavily into this, but the interesting piece from our side is that we have this research analytics database here with a number of projects hung off it, one of which is the Future MS project. We want the users here to be able to ultimately see the data as it appears within the NHS health boards off their SciStore database in near real time, pulling that in with the sequencing data that's coming from my own lab and the CRF data that's coming from the MS clinics. That's the aim, if you like. We have spent a lot of time working with the health leads from those 4 NHS boards in the early stages of the project planning. And we've come up with, effectively, a technical solution that allows us to effectively automate the retrieval of those nine data fields from the consented patients. We obviously only want those data for the patients who are consented, and there'll be many other patients within those health boards who've got the same data set, but we don't want theirs. We'd like to get the data in near real time. Clearly, we don't need it 5 minutes afterwards, after the data was generated, but we certainly don't want it 5 months after it. The solution needs to work across multiple health boards. And what we wanted to develop here was not just a tool that worked on the future MS study, but have a tool set that work on any future study, because effectively what we want to do is just redefine what the data sets we need are. And then the same tool should work again for oncology projects or rheumatoid arthritis projects or anything else we want to do. And the health leads were very keen to support that because they have the same issue themselves, and they keep getting these crazy requests from researchers to do all this stuff, and they haven't got the resource to do it. So if we can actually build a platform that does it, that's going to help. And so this needed to have engagement and buy-in from the health leads. So effectively what we've got here is a capability, I mentioned earlier, we have a patient ECRF that will obviously hold the names and the personal data and the KI numbers of the patients who consent to the study. What this platform will do is generate a CSV file, comma separated value file of that data set as it builds, and make that available through a secure file transfer protocol to an Ensemble integration engine within the NHS N3 firewall that will then query for those KI numbers for a defined time-based patient arrived after this date, and between now and the particular field values that we request to those source systems, pull it back in, and then make that available through an inbound STP endpoint to our clear clinical database, which will do some sanity checks around have I got the right number of data fields back from what I asked for, have I got the right number of patient values back from what I asked. And then there'll be a de-identification capability that then ports that into the research analytics piece that then allows the scientists working on the project to see the phenotype and genotype data and undertake the analyses and phenotype-genotype comparisons they want. So that's what we're putting together. That's a pretty version of the same that basically just shows that you've got this internal piece with the Ensemble integration engine that works across the 4 health boards, have clones of that themselves. So that's all going together as we speak. In terms of summary, what we want to have here is the ability to reach in and have an interoperable system with the NHS to get consented data into our systems in a near real-time basis. We want it to work across multiple health boards because obviously you don't know which health board you're going to be working with. It'll support our research projects. The hope is that it will also support other projects across NHS industry within NHS Scotland. We want it to have applicability for future projects going forward. And I think if you like the learning message from this, if you like the one take-home message, is don't start thinking about this when the contract's signed. Start thinking about this 12 months before you get that far so that you've got buy-in from the NHS e-health leads. They're the people who have to implement this, make it happen. They've got significant amounts of influencing and buy-in to achieve in their own worlds. So don't just arrive at the last minute and say, can you sign up to doing this and tell me how much it'll take to do it? Because it takes quite a long time for them to work this out. But we think we've engineered a very neat technical solution here. And over the next 2 years, we're going to be populating this out. And hopefully we'll be coming back in a couple of years' time to tell you how it's all worked. Recognise that this is a cast of thousands behind the scenes. My own team in Glasgow, who are running the core facility, as well as the 4 universities and the 4 NHS boards across Scotland who are forming the backbone of this project, as well as our 2 industrial partners, Thermo Fisher and Iridia. Happy to take any questions. Thank you very much.